In the realm of biology, few organs can claim the regenerative prowess of the human liver. Unlike most organs, which are limited in their ability to repair themselves after damage, the liver possesses a unique capacity to not only heal, but to regenerate and restore itself to full function even after significant portions have been removed or destroyed. This extraordinary ability is not just a fascinating biological phenomenon, but has profound implications for medicine, particularly in the fields of surgery and transplantation. In this in-depth exploration, we will delve into the complex mechanisms that allow the liver to regenerate, the role of various cells and signaling pathways, the influence of the gut microbiome, and the clinical implications of this regenerative capacity. The liver is a large, reddish-brown organ located in the upper right side of the abdomen. It is divided into two main lobes and is the largest internal organ in the human body, weighing around 3 pounds in adults. The liver is responsible for a wide array of critical functions that are vital for maintaining overall health. These functions include detoxifying harmful substances, producing bile to aid in digestion, storing essential vitamins and minerals, regulating blood clotting, and metabolizing nutrients such as carbohydrates, proteins, and fats. The liver's ability to perform these diverse functions is made possible by its highly organized structure, which includes millions of specialized cells called hepatocytes. Hepatocytes account for about 80% of the liver's mass and are the primary cells involved in its metabolic activities. These cells are organized into lobules, which are the functional units of the liver. Blood flows through these lobules, allowing hepatocytes to filter and process the blood, removing toxins and waste products. Given its essential role in maintaining homeostasis, the liver's ability to regenerate is a key factor in its ability to recover from injury or disease. Unlike other organs, where cell death often leads to irreversible damage and scar tissue formation, the liver can regenerate lost tissue and restore its full range of functions, albeit with some limitations. The process of liver regeneration is a complex and finely tuned response to injury or partial hepatectomy, surgical removal of a portion of the liver. This regenerative process involves the proliferation of hepatocytes, as well as other non-parenchymal cells, and is regulated by a variety of growth factors, cytokines, and signaling pathways. Hepatocytes, the primary functional cells of the liver, are at the forefront of the regenerative process. When the liver is damaged or a portion is removed, hepatocytes are triggered to exit their usually quiescent, inactive state and enter the cell cycle, leading to cell division and proliferation. This response is known as compensatory hyperplasia, a term that reflects the liver's ability to increase the number of cells to compensate for the loss of tissue. Several key growth factors play a pivotal role in initiating and sustaining hepatocyte proliferation. One of the most important is hepatocyte growth factor, HGF, which is produced by non-parenchymal cells in the liver as well as by other tissues. HGF binds to its receptor, c-Met, on the surface of hepatocytes, activating a signaling cascade that promotes cell proliferation, migration, and survival. HGF is particularly crucial in the early stages of liver regeneration, helping to kickstart the process of tissue restoration. Another critical factor is transforming growth factor alpha, TGF-alpha, which, like HGF, stimulates hepatocyte proliferation through its interaction with the epidermal growth factor receptor, EGFR. TGF-alpha is thought to work in concert with HGF to ensure that hepatocytes proliferate at a rate that matches the body's needs. Interestingly, while these factors promote cell division, other molecules, such as transforming growth factor beta, TGF-beta, act to inhibit hepatocyte proliferation once the liver has regained sufficient mass. This negative feedback mechanism is essential to prevent uncontrolled growth and the development of liver tumors, underscoring the highly regulated nature of liver regeneration. While hepatocytes are the stars of the show, non-parenchymal cells also play critical roles in liver regeneration. These include endothelial cells, Kupffer cells, resident liver macrophages, and hepatic stellate cells. The liver's blood vessels are lined with endothelial cells, which are essential for maintaining the organ's blood supply. After liver injury or partial hepatectomy, the liver experiences changes in blood flow, which in turn stimulates endothelial cells to produce various growth factors, including HGF and vascular endothelial growth factor, VEGF. VEGF is particularly important for angiogenesis, the formation of new blood vessels, which is crucial for supplying oxygen and nutrients to regenerating liver tissue. Without adequate angiogenesis, the newly formed tissue would be deprived of the resources it needs to survive and function. Kupffer cells are specialized macrophages that reside in the liver and play a key role in the immune response. During liver regeneration, Kupffer cells are activated by signals from damaged tissue and begin to secrete cytokines, such as tumor necrosis factor alpha, TNF-alpha, and interleukin-6 IL-6. These cytokines have multiple effects, including promoting hepatocyte proliferation and modulating the inflammatory response, which is essential for clearing damaged cells and preventing infection. Hepatic stellate cells are involved in the production of extracellular matrix components, which provide structural support to the liver. Under normal conditions, these cells are quiescent, but they can become activated in response to liver injury. Once activated, hepatic stellate cells produce factors that can either support or inhibit liver regeneration, depending on the context. In cases of chronic liver injury, excessive activation of hepatic stellate cells can lead to fibrosis, the excessive accumulation of scar tissue, which can impair liver function and limit its regenerative capacity. In recent years, researchers have discovered that the gut microbiome, the complex community of bacteria, viruses, fungi, and other microorganisms living in the intestines, plays a significant role in liver regeneration. This connection, known as the gut-liver axis, is facilitated by the portal vein, which carries nutrient-rich blood from the digestive tract directly to the liver. The gut microbiome influences liver regeneration through several mechanisms, primarily by producing metabolites that can either promote or inhibit the regenerative process. For example, certain gut bacteria produce short-chain fatty acids, SCFAs, such as butyrate, propionate, and acetate, which have anti-inflammatory properties and can enhance hepatocyte proliferation. These SCFAs help create a favorable environment for liver regeneration by modulating the immune response and promoting tissue repair. On the other hand, some gut bacteria produce lipopolysaccharides, LPS, which are components of the outer membrane of gram-negative bacteria. LPS can trigger an inflammatory response in the liver by activating toll-like receptors, TLRs, on Kupffer cells, leading to the production of pro-inflammatory cytokines. While a certain level of inflammation is necessary for tissue repair, excessive or prolonged inflammation can impair liver regeneration and contribute to liver disease. The balance between beneficial and harmful bacteria in the gut is therefore crucial for optimal liver regeneration. Factors that disrupt this balance, such as poor diet, antibiotic use, or gastrointestinal diseases, can have a negative impact on liver health and its ability to recover from injury. This understanding has led to growing interest in exploring how interventions that modulate the gut microbiome, such as probiotics, prebiotics, and dietary changes, could support liver regeneration and improve outcomes in patients with liver disease. The liver's regenerative capacity has significant implications for clinical practice, particularly in the context of liver transplantation. For patients with end-stage liver disease or acute liver failure, transplantation is often the only viable treatment option. However, the limited availability of donor organs has led to the development of techniques that take advantage of the liver's regenerative abilities, such as partial liver transplantation and living donor liver transplantation. Partial liver transplantation. In partial liver transplantation, only a portion of a donor liver is transplanted into the recipient. This approach is possible because the transplanted liver segment can regenerate to meet the metabolic demands of the recipient's body. Partial liver transplantation is especially useful in pediatric patients who require smaller liver grafts and in cases where a full liver graft is not available. The success of this procedure depends on the ability of both the donor's and recipient's livers to regenerate and restore full function. Living donor liver transplantation. In living donor liver transplantation, a healthy individual donates a portion of their liver to a recipient in need. Both the donor's remaining liver and the transplanted liver segment regenerate, allowing both individuals to recover with fully functional livers. This procedure has several advantages, including reducing waiting times for recipients and allowing the surgery to be planned electively. However, it also carries risks for the donor, and careful selection and evaluation of potential donors are critical to ensure a successful outcome. Regenerative medicine. Advances in regenerative medicine and tissue engineering hold the potential to further leverage the liver's regenerative capacity for therapeutic purposes. Researchers are exploring the use of stem cells, bioengineered scaffolds, and gene therapy to enhance liver regeneration in patients with chronic liver disease or after major liver resection. For example, stem cells derived from the patient's own body or from a compatible donor could be used to regenerate liver tissue, potentially reducing the need for transplantation. Another promising area of research involves the development of artificial livers or bioengineered liver tissues that could temporarily support liver function in patients with acute liver failure, giving the native liver time to regenerate. While these approaches are still in the experimental stage, they offer hope for new treatments that could improve survival and quality of life for patients with liver disease. Despite the liver's remarkable ability to regenerate, there are limitations and challenges that can affect the outcome of the regenerative process. Chronic liver diseases such as cirrhosis, hepatitis, and non-alcoholic fatty liver disease, NAFLD, can severely impair the liver's regenerative capacity. In these conditions, ongoing damage to liver tissue leads to the accumulation of scar tissue, fibrosis, which disrupts the liver's normal architecture and impairs its function. In cases of cirrhosis, where fibrosis is extensive, the liver's ability to regenerate is significantly diminished. This is because the formation of scar tissue interferes with the proliferation of hepatocytes and the organization of regenerating tissue into functional lobules. As a result, even if the liver regenerates to some extent, the new tissue may not be fully functional, leading to progressive liver failure. Another challenge is that while the liver can regenerate its size, the new tissue may not be identical to the original. Structural changes in the liver, such as altered lobule architecture or abnormal blood vessel formation, can affect the liver's ability to perform certain functions. These changes are particularly relevant in the context of liver transplantation, where the success of the procedure depends on the newly regenerated liver tissue being able to meet the metabolic demands of the recipient's body. Moreover, the regenerative process itself is subject to tight regulation to prevent uncontrolled growth and the development of liver tumors. In some cases, mutations or disruptions in the signaling pathways that control liver regeneration can lead to the formation of liver cancer, such as hepatocellular carcinoma, HCC. Understanding the balance between promoting liver regeneration and preventing tumor formation is a key area of ongoing research. The liver's ability to regenerate is one of the most remarkable examples of the body's capacity to heal and adapt to injury. This regenerative capacity not only underscores the liver's vital role in maintaining overall health, but also offers hope for new therapeutic approaches to treat liver disease and extend the lives of patients with severe liver conditions. As our understanding of the mechanisms underlying liver regeneration deepens, new opportunities for regenerative medicine and transplantation are likely to emerge. Whether through the use of stem cells, bioengineered tissues, or interventions that modulate the gut microbiome, the future of liver medicine holds the promise of harnessing the liver's natural superpower to improve health outcomes and offer new hope to those affected by liver disease. In the meantime, the liver remains a testament to the resilience and adaptability of the human body, a true biological marvel that continues to inspire scientists, clinicians, and patients alike. As we continue to explore and unlock the secrets of liver regeneration, we may one day fully harness this extraordinary ability to heal and regenerate, leading to new treatments and therapies that could transform the way we care for the liver and the people who depend on it. And that's a wrap on our deep dive into the liver's incredible ability to regenerate. If you found this exploration of one of the body's most fascinating organs as mind-blowing as I did, don't forget to hit that like button and subscribe for more content like this. And as always, thanks for watching. Stay curious, stay informed, and I'll catch you in the next video.